So here are my disclosures. I think everyone knows who I am. I'll give you a little brief history. So I'm a virologist by training, done gene therapy, AAV gene therapy for more than 30 years, so I'm much older than I look. And I'm only at the NPS Society for a year and a half, so this is relatively new to me, although I have worked with Doug and Hyann on their Abiona 3A and 3B programs, so I'm familiar with that. But I want to just give an overview of gene therapy in general, what works and what doesn't work, and my vision for the future. So current treatments available are ERTs, so we welcome the approval of Avlea that crosses the blood-brain barrier, but there's a number of enzymes that provide benefit to the body, but less so to the brain and other hard-to-reach tissues. Hematopoietic stem cell transplant is a good treatment. It's been around for a long time, but of course it's not perfect. None of these therapies are perfect, and so we need something better. We've needed something better for a long time. So why do we need gene therapies and what makes them better? So ERTs are life-saving, but they're expensive. They require weekly infusions. Patients have to travel. It's very time-consuming. And the ERTs, most of the ERTs currently don't reach the brain. HSCT is life-saving, one-time treatment, but there is a significant risk of the toxic conditioning. So these kids have to undergo very serious conditioning regimen. There's roughly a 90% survival, and it may not correct the secondary musculoskeletal disorders. The beauty of gene therapy, which is why I went into the field, it offers a one-time, lifelong, durable treatment option. It can reach the central nervous system where the enzymes typically do not reach. And early treatment, as we've seen with many of the therapies, early treatment is the best option. So what are the different types of gene therapies? So they typically use a virus to deliver the corrected gene cargo. So you've got adenoviruses, which are large, but they have immune responses, so they're less preferred. We had some early patient death back in the late 90s. We switched to two other vector delivery systems, adeno-associated viruses, which are small. They have an unfortunate physical limit of the payload that they can deliver. They can only deliver roughly 4 kb of genetic material. And then we have lentiviruses. The beauty of adeno-associated viruses is they don't cause any disease. They're a natural virus. They don't cause any disease in humans. Whereas lentiviruses, they're completely gutted because they do cause disease in humans, as we well know. So gene therapy, it's delivered directly into the body. So AAV, it's very small, as I said, doesn't cause disease. But the problem is, once you get it, you can't redose. So most people, they develop antibodies that prevent redosing. There's also a lot of seroprevalence of circulating serotypes, and if you've been exposed as a child or an adult to a circulating serotype, that could prevent you from being treated in a gene therapy protocol or an approved gene therapy because you develop neutralizing antibodies which would block the effect. AAVs don't readily integrate in human cells, which is a good thing, whereas lentiviruses do integrate, so you've got to titrate the amount of vector copies that you can deliver on a per-cell basis. AAVs can remain active for the life of this cell. They basically survive as an episome in the nucleus, so it doesn't integrate. And AAVs can also be modified to improve the delivery to hard-to-reach tissues. And some of that is shown in the red images on the figure here. So we can modify them And a number of companies have modified them. There's also some natural serotypes that you'll hear more about in the next year or two that greatly improve the ability to deliver this genetic cargo to hard-to-reach tissues. So adeno-associated virus, let's see, can we play the movie?
More than 30 million Americans suffer from rare diseases, most caused by a single defective gene. Each unique rare disease can affect as many as 200,000 patients, or merely a few dozen. For many rare diseases, there are no FDA-approved treatments, meaning patients and their families are left to simply endure challenging and often grievous symptoms without hope for a cure. The Bespoke Gene Therapy Consortium, managed by the Foundation for the National Institutes of Health, is leading the charge to change that, with an exciting new type of gene therapy bringing hope to rare disease patients, adeno-associated virus technology, or AAV. So how does it work? First, it's important to remember that viruses are made of genetic material and enter the cells of other living organisms to reproduce. We often think of viruses as causing illness, like the common cold. But adeno-associated viruses are special. Scientists can engineer them as delivery vehicles that carry therapeutic genes to cells affected by rare genetic disease without causing illness. Often, patients suffering from rare diseases are not producing critical proteins, and these therapeutic genes contain the instructions to create them. The therapeutic gene produces more protein to replace the missing or non-functioning proteins in the tissues and organs affected by rare disease, addressing the root cause of the disease. This approach is emerging as one of the most promising paths to treat rare genetic disorders.
So this video was put together by the Bespoke Gene Therapy Consortium, and they're an organization that is developing a gene therapy for MPS IVA, and we're currently working with that program. So how do we make AAV? So the wild-type AAV genome looks like this. As Eleni mentioned previously, it's flanked by these inverted terminal repeats. So to make an AAV vector, we remove all of the internal normal virus regions, so the rep and the cap genes, and replace them with your therapeutic gene. This is put into a plasmid, which encodes this vector genome. We also deliver two other plasmids, an AAV helper and an ad helper. So the AAV provides the rep and cap genes that you want to package the vector into, and the ad helper just provides the help to get this whole thing assembled. You then lyse cells, and you harvest your AAV virions, and you purify them away, and that becomes your drug product. So AAVs are good, but they're not perfect. They're stable viruses. They last a long time in cells without integrating. Between 20 and 50% of the people may have high antibody levels, which prevents them from being treated. The majority of the virus goes to the liver in most serotypes. So to get around that, that's if you deliver it intravenously. If you need brain delivery, you need to deliver it directly into the spinal cord or the spinal fluid. Unless you have one of these latest capsids that can cross the blood-brain barrier, either via transferrin receptor modality or other new novel serotypes that you'll be hearing about more in the future. It's a low infection rate per cell, so you might require 10 to 100 particles per cell to get a good transduction of that cell. It requires a lot of virus to get an efficient delivery treatment, and it's very challenging to make a large amount of virus. To go into a patient, you need trillions of vector particles to get an efficient delivery via IV. The expression only lasts as long as the life of the cell. So if most of that virus goes to the liver turns over regularly. So it's not going to last more than probably a decade unless you get a random integration event. So how can we make this better? Or let's switch to ex vivo gene therapy. So like the MPS IIIC that Rafaela is developing, This is an ex vivo therapy. He already talked about how that's produced. You harvest stem cells, transduce them in culture with the lentiviral vector, expand them, purify them, and the patient undergoes a conditioning regimen, which is toxic, but then you deliver those stem cells that have been treated back into the patient, and hopefully that engrafts, and the patient's own body becomes the biofactory for that gene therapy. The autologous HSCT, that's Raphael's program, that's the most effective because you have a lot less graft versus host disease, which is a serious concern as far as HSCT. Let's see, I don't need to go through, well, so the HSCT, the goal is to treat the cells, and those cells then spread throughout the body, and each of those depots of cells become the cell factories or enzyme factories for their gene therapy product. But lentiviruses are good, but they're not perfect. It's stable integration, so the stem cells grow and divide into other cell types of blood cells and produce your therapeutic gene. But some donor cells as a recipient, so there's no graft versus host disease. You may get cognitive benefit if enough cells cross the blood-brain barrier. There's a difficulty in reaching tissues like the eye and bone. We see that in many gene therapy protocols. Again, the toxic conditioning regimen, you can get a 90% survival, less so if it's allogenic. The initial cell transfer may not engraft, and it requires additional transfers, and it may not correct the musculoskeletal disorders. It's time-consuming to produce, and like most gene therapies, it's very expensive. So another novel cell therapy treatment is being developed by Immusoft, and we heard their talk earlier. So that's utilizing a B cell therapy. So the goal there is to collect and purify your B cells. They do a non-viral delivery of mRNA and the gene of interest into these cells. The transposon, which comes from the mRNA, it integrates the gene of interest into the B cells. The B cells expand. They differentiate into protein factories, just like your antibody-producing cells. They pump out the missing protein. You get stable expression for the life of the cell, and as was mentioned earlier, it can be re-administered. So that's a really impressive property of this Immusoft therapy. There is still a potential risk of random integration, so they have to reduce the vector copy number per cell, and there's an unknown durability depending on the cell types that are transduced. So gene therapy programs for MPS and ML. Currently, we know about many of these. REGENXBIO with their MPS I and MPS II programs. We hope to get approvals sometime soon for the MPS II program. Ultragenyx, hopefully in a month and a half, we'll see an approval for the 3A program. These are utilizing AAV9 gene therapy vectors, which we know cross the blood-brain barrier to a degree, but not as much as we would like, ideally. Orchard Therapeutics, their MPS I is in phase 3, and their 3A product is preclinical, and that's a lenti-HSCT treatment. I talked about the Immunosoft MPS I and MPS II programs. The MPS II is in preclinical development, and that's the B-cell autologous cell therapy. University of Manchester, they have the MPS II program. It's a lenti-HSCT, and that's a phase 1/2 program. And in the future, Patty Dixon at WashU, as we heard earlier, she's developing the ML2, ML3 program. That's going to be an AAV-based approach, and that's going to hopefully use a novel capsid that can deliver the cargo to the hard-to-reach tissues of the heart, of the brain, and also of the bone. And Francois Piguet, we're working with to develop the 3B program. That's also going to use a very novel serotype capsid that's never been tested in humans. and that's in preclinical development, and hopefully we'll be going into the clinic next year. The blood-brain barrier crossing ERT therapy programs, we know Denali, JCR Pharmaceuticals, those are very impressive technologies that can be leveraged for many other MPS conditions. The Spruce program, it's not blood-brain barrier because they use an Ommaya reservoir to deliver the enzyme directly into the CNS, and that's licensed by Spruce. And so how can we build a better gene therapy? The idea would be something that can treat hard-to-reach tissues like the brain, the bone, and the eye, something that gives you a long-lasting, durable expression, something that gives you lower doses that are required to get an effective treatment, because with some of the AAV programs, if you've got to go in with 10 to the 14th vector genomes per kg in a small child or even an adult, that's a lot of virus. It's difficult to manufacture. It's very expensive. Ideally, you want something that's less immunogenic and can be redosable, like the Immunosoft program. And you want something that can be simplified in their manufacturing. A lot of the cost in the development process is manufacturing, and that has to be recovered for every patient treated. So we greatly need to reduce the cost for these rare and ultra-rare diseases so that we can deliver therapies not only to well-to-do countries like the United States and Europe, but throughout the world. So I've already talked about some of the issues in blood-brain barrier delivery. It requires either a complex surgery, poor diffusion of the vector, intrathecal delivery, or cisterna magna delivery. It's simple administration, but it's very challenging for the patients and for the developers to develop these therapies. AV9 that does cross the blood-brain barrier currently, you get maybe less than 1% of CNS transduction with an AV9. So that's why the newer capsids that cross the blood-brain barrier more efficiently are the future for AAV gene therapy. There's also other delivery issues. If you need to treat musculoskeletal and cardiac delivery, you can either do direct injection into the muscle, but that's not very efficient. It's hard to treat. If you do IV injection, you only get a moderate efficiency to skeletal and cardiac muscles, depending on the serotype. AV9 biodistribution, 90% of that virus goes to the liver, 3% of the spleen, lymph node, heart is at 2%, muscle is less than 2%, so it's not very efficient to these hard-to-reach tissues. So again, I talked about how you improve the delivery. You alter the structure of the capsid, and a lot of companies are using AI to rationally design and select capsids that have much improved tropism to these hard-to-reach tissues like the retina, eye, brain. It also improves the manufacturability. So these AI systems select variants that are much easier to manufacture and much cheaper in the long run. And they also develop variants that have reduced neutralization by pre-existing antibodies. So the goal would be to be able to treat many more patients that normally wouldn't be able to be treated. So companies like Dino, 4DMT, Capsida, Voyager, and others are licensing these improved capsids to large pharma. So how do we improve the durability of gene expression? So again, if we improve the tissue targeting, we need the virus to stick around longer. So you want lower doses of the vector to improve the targeting. If you have higher doses of vector, you're going to get a larger immune response. It's not going to last as long. Target tissues. So if your tissue is a rapidly dividing cell like the liver, it's going to turn over. So you want to go into areas that don't turn over, like the CNS or muscle, so that your gene expression lasts for a longer period of time. We can develop an improved immune modulation system for systemic administration, and in the future, that might actually allow re-administration of the vector. If you can stop or blunt the initial immune response or do apheresis to remove the pre-existing antibodies, you might be able to redose some patients. So what's the future of gene therapy development, of AAV gene therapy treatments? Again, develop modified capsids that efficiently target the CNS or the muscle or bone and eye for a more durable gene expression. You want to generate a stable vector production system, so a master cell bank that can rapidly switch between different gene of interests, a vector-containing gene of interest delivered via either a nanoplasmid or a minicircle. I didn't talk about some of those components, but it removes the unwanted genetic material. So you're not delivering anything that you don't need to the patient. You only deliver the genetic cargo. You can electroporate a minicircle into a master cell bank, or you can use new treatments like CRISPR or transposon integration into your master cell bank. And you can freeze down these transfected cells as part of a working cell bank. All of these are short circuits to streamline that manufacturing process, reduce the cost of delivery so that your therapy doesn't cost $3 million per patient. If you can reduce that by a factor of 10, you'd be able to reach many more patients. The future of cell therapy treatments. Ideally, if you could develop a point-of-care process that can deliver a therapeutic gene into hematopoietic cells, if you can shorten the time frame for gene delivery and infusion into cells, ideally in a single day, you want to simplify the delivery and integration of the therapeutic gene into the cells, and there's mechanisms today that we could do that. You want to minimize the patient risk and hospitalization burden before and after the treatment. Right now, these treatments take weeks to develop. Patients have to undergo they have to stay local for several weeks to get this treatment. The cells have to be harvested, purified, tested, frozen, shipped, re-administered. So we want a method that can reduce all that cost and burden on the care. And you want to increase the number of treatable conditions, especially ultra-rare conditions, because current treatment strategies, they're not really tenable for future ultra-rare conditions. Biotech companies are not going to spend $100 million, $200 million developing something that's going to treat 20 kids a year, especially in third world underdeveloped countries. So we need a better system. That's my goal. Hopefully we can develop that with the people in this room and our combined forces in the very near future. I hope we all share that goal. Safer, more effective treatments, and I want to thank you. Questions? I can't run around with the microphone. She's coming. Oh,
Thank you.
Joe.
As you appreciate, there's lots of challenges. Have any of these companies that are developing the newer, I'll call it second-generation capsids, which are much more efficient, have they gone to clinical trials yet, any of them?
Is this on? Yes. Capsida is one company that has developed a novel capsid that was tested in patients. Unfortunately, we don't know what happened, but that first patient that was treated passed away. That's still under evaluation. There's not much information that's been released from that company, but there's many other companies that are currently doing their pre-IND work, and more of these therapies will go into patients soon.
And you're going to announce your 3B company in sometime soon?
Yes, that's undergoing licensing agreements as we speak, but nothing can be disclosed at this point. But you will be amazed at this capsid and some of the ability of this capsid not only to transduce these hard-to-reach tissues, but the novel ability of no immune response to this capsid. So, more to come. I can't disclose anything else, or Francois will kill me. It's hard to see. Did you have a question? No. Questions? Nothing? All right, well, thank you all very much. This has been a wonderful conference. I hope to have much more news next year that we can present on these clinical studies and fundraising for the ML program. That's a key priority of the society right now. And thank you.